Hello students, in this video we are going to see about morphogen and morphogenic gradient under the topic of Developmental Biology Unit 5 Iicsirnet Life Sciences portion. Okay, we are going to see about what is morphogen and what is morphogenic gradient. First up, we can see about what is morphogen. So what is morphogen? Morphogen can be any substances. Morphogen can be signaling molecules. Morphogens can be transcription factors. They can be paracrine signals. We have already seen about this paracrine signaling when we spoke about instructive interaction, when we spoke about cell-fate attainment of stem cell. It helps in cell-fate attainment in stem cell. They work, how do they work? They work on concentration dependent manner. They help in conditional specification. We have already uploaded a video based on the definitions and the terminologies of specification, where we spoke about this term called conditional specification. So this morphogen help in attainment of conditional specification. These morphogens are diffusible. This is an important characteristic of morphogen. Only then these substances can diffuse from one medium into the cell, and it helps in attaining specific fate of the cell. Okay, these are the basic characteristics of morphogen. They work in concentration dependent manner. These morphogens work in concentration dependent manner. So these are the basic properties. These are the basic properties of morphogen. Signaling, transcription factor, paracrine signaling, diffusible molecule, help in attaining cell fate. They are concentration dependent. They work on concentration dependent manner. They help in attaining conditional specification. These are the basic properties of morphogen. Okay. So how do these morphogens work? Consider in case of C. elegans. In case of C. elegans, we will be seeing this in detail when we speak about C. elegans. We have an anchor cell. We have other cells. Vulval precursor cell. P3P, P4P. We have other cells. P3P, P4P. P5P, we have P6P, P7P, P8P. Okay. Consider this every cell as a vulval precursor cell. We are taking an example of C. elegans and we are trying to explain what is morphogenic gradient. This is an anchor cell. Okay. This anchor cell will give some signals. We will speak about what are the signals and how these signals are derived. So this anchor cell will give rise to so many signals. These are morphogenic signals. This P6P will attain the primary cell fate, because it receives majority of the concentration of the morphogen released by this anchor cell. So this P6P will become primary cell fate, primary precursor cell, and the adjacent will become secondary cell because they receive only less amount of morphogen. This length 3. Length 3 is the signal released by the anchor cell here. P5P and P6P will attain secondary fate, whereas P5P and P7P will attain secondary fate, whereas this P6P will attain primary fate, because majority of the morphogen released by this anchor cell will reach this P6P. Since high concentration of the LEN3 reaches this P6P, this P6P will attain primary fate, and P5P and P7P which receive comparatively low amount of LEN3 that are produced by this anchor cell will give rise to secondary fates. Okay? This is an most important example of morphogen and morphogenic gradient. If the concentration of receiving this morphogen differs, so it differentiates into primary and secondary. There is another reason also of producing LIN12, which prevents it from attaining primary fate. This is another reason for P5P and P7P for not attaining this primary fate. In addition to this morphogenic gradient is also another example. This is different story. This lateral inhibition, this red ink, lateral inhibition is different story, but this morphogenic gradient is also an interesting example of explaining why P5P and P7P are not up to the level of primary cell fate, and P5 and P6P in attaining the primary cell fate because it receives large amount of morphogens that are released by this anchor cell. Hope I have explained about morphogenic gradient and concentration of morphogenic gradient. What will be the result of the cell fate based on the change in the concentration gradient of morphogen that is released by one cell. Next we will see a graph on x-axis and y-axis. On x-axis we have morphogen. Sorry, very sorry for that. Y-axis, x-axis. X-axis we have distance, and y-axis we have morphogen. So consider a graph like this. Threshold 1, threshold 2. Consider this as a cell. Sender as a inducer cell that gives out morphogen. This is a receiver cell. This concentration gradient should be above this threshold level. Only then it can reach the receiver and it can give rise to a specified and desired cell fate by this receiver. So this threshold level should be crossed by the morphogenic gradient, threshold 1 and threshold 2. Consider the true threshold level. This morphogen should cross this threshold level. Only then, these are the two threshold levels. This morphogen should cross these threshold levels. Only then it can give rise to desired function in the receiver end. This is the basic theory. This is the basic threshold theory with regard to morphogenic gradient. So it should come across this threshold level. Only then it can help the receiver to attain specified fate. Explaining this in simple terms. Okay. For example, to clear the CSIR exam, you should study. You should collect materials. One student should read about the previous year questions. These things are hard things to do. for any PhD aspirant to clear CSIR exam. This is a threshold level. If you cross this threshold level, there are high chances for you to join PhD in well-reputed institute. So this is a threshold. Any student who are preparing for this exam should cross this threshold level. This is the basic threshold. Your hard work should help in pursuing all these, clicking all these check boxes. Only then you can cross this threshold level to become a PhD scholar in some good institution. So this is the threshold. Likewise, Morphogen is also having a threshold level where we have seen that distance, Morphogen and a graph explaining threshold. Threshold 1. So, your Morphogen should cross this threshold, only then it can give rise to a specified self-weight in the receiver end. Okay. This threshold level crossing is based on the concentration of the Morphogen. So it is called as morphogenic gradient. Summing up all the things that we have seen in the short video, we have seen about the definition of Morphogen. What are the definitions of Morphogen? We have seen that Morphogen is a transcription factor, Morphogen is a paracrine factor, Morphogen help in attaining self-weight in conditional and regulative development. It is a diffusible molecule, it is a signaling molecule. So, we have explained with the help of C. elegans, we have explained how P6P attained primary self-weight, P5P and P7P reaches secondary self-weight. We have explained this with an example as well. Then we have seen this graph-oriented threshold. Threshold concept of morphogenic gradient. Hope this video is very clear and it help in basic understanding of forthcoming videos that will be on chick development, in Drosophila development, in tetrapod lymph development and all those things. So Morphogen is a basic component in developmental biology. Hope this Morphogen content is very clear. Thank you for watching this video. Bye.